Hello, my name is Rachel Seo, and in this video, I'll be discussing the following New York Times article, How Boa Constrictors Breathe While Squeezing the Life Out of Their Prey, as well as the corresponding primary source article, Modular Lung Ventilation in Boa Constrictor. For this assignment, I'll determine if the New York Times article accurately displays the findings in the primary source article. The New York Times had published the article on March 22, 2022, discussing the findings in the Journal of Experimental Biology of how the boa constrictor got its famous name. The New York Times article follows the results of Elizabeth Brainard's monumental study, Modular Lung Ventilation in Boa Constrictor. According to the researchers, it's been shown that boas have evolved their bodily functions, such as the ventilation system, in order to maneuver their body to function in a specific type of way. By boa constrictors having an evolutionary advantage digesting and ingesting their prey by modifying their lung ventilation, as the study has been found, they can remain a competitive predator in their natural habitat. Firstly, the researchers had proposed a theory. When constricting and ingesting prey, snakes are most likely changing the specific region of their ribcage that's expanding, but another option would be that they use any uncompressed area of the ribcage to draw air into their lungs. Now, in order to test this hypothesis, a group of scientists visualized a boa constrictor's ribcage during constriction using 3D ray technology. Once the snakes had received anesthesia, the team implanted metal markers no bigger than 0.5 millimeter in the ribs and vertebrae they wanted to image. Then, they filmed these regions with X-ray video. Using a blood pressure cuff, they restricted the boa's rib motion in specific regions, simulating what happens in nature when these snakes constrict their prey. They had discovered that when resting, boa constrictors breathe using ribs near the upper third of their lungs. But when the blood pressure cuff was wrapped around those ribs, a specific set of ribs further down the snake's body began expanding to draw in air. According to the researchers, the snake just turns off a section of the ribcage and then turns on another section, says Dr. Capano, who also adds that as soon as the cuff was removed, the ribs used to breathe during rest immediately re-engaged. According to the New York Times, it is believed that this ability to modulate rib engagement emerged while or before snakes evolved the ability to constrict, and perhaps ahead of a snake's ability to eat large prey. Without this breathing adaptation, they say that snakes may not have become such a diverse class of reptiles spread out across the globe. In this primary source, Modular Lung Ventilation in Boa Constrictor, there had been reported findings behind the evolution of constriction and of large prey ingestion within snakes, discovering that there are key innovations that may explain the remarkable diversity, distribution, and ecological scope of this clade relative to other elongate vertebrates. However, based on the results, these behaviors may have simultaneously hindered lung ventilation, such as that early snakes may have had to circumvent these mechanical constraints before these behaviors could evolve. To begin this experiment, researchers used 3 adult female boa constrictors that varied from large to moderately sized. There were 5 separate boa constrictors used for the electromyography and videography portion of the study conducted at Dickinson College. For electromyography, they used an adult female and male. One individual used for the Brown University portion of this experiment was acquired from a reputable reptile breeder. The remainder of the animals were captive-born snakes bred from individuals wild-caught in Belize, Central America. The snakes were maintained in a climate-controlled animal care facility at 60% relative humidity and were kept on a diet of dead rats. In the beginning of the trials, pneumotachograph was used to measure airflow when the snakes were consuming food. Electromyography was used in 2 boas to quantify whether the lung capacity was capable of spatially discrete patterns of activation. A handful of trials were simultaneously recorded with light video cameras via GoPro at 25 frames and intermittently synced through inclusion of EMG signal timestamps into the video recordings. All of the data was collected within 7 days of electrode implantation and EMG signals. The timestamps of EMG and video recordings were to associate muscle activity with externally observable lung ventilation motions. During constriction, we found that the boas ventilated in trunk segments, meaning that the ribs rotate during lung ventilation. Here is why the New York Times article is proven to be accurate when looking at the primary source. This is proven through these 3 points. To wrap up this discussion, it is evaluated how both sources reflect off of each other's work, showing that the data collected from each source can be perceived as a highly relied on source of information.